After discussing the isolation and then pure culturing and then screening, after the screening, when we came to know that this particular isolate or an organism is our choice and can meet all those characteristics by which we can produce our desired product, then the step is that we have to identify that organism. That organisms, which phyla, which group, which genus and which species belongs to that. So for this purpose, there are various techniques are employed to characterize and to identify that screened organism. The first choice is the phenotypic characteristics. Use of the phenotypic characteristics is a very conventional approach for the identification, in which we use the microscopic morphology, metabolic differences, which we called as biochemical testing, biochemical characterization. On the other hand, we can say that physiological and biochemical characterization, serology, fatty acids analysis. So these are different aspects which we can identify and note while identifying any organisms. So this is known as a conventional approach. But now in modern age, the genotypic characteristics are very helpful and a rapid method of identification, in which we use different nucleic acid probes, DNA amplification and ribotyping. So along with and the mixing of these two, either the phenotypic characteristics as well as the genotypic characteristics, it is very precise and ultimate method of the identification of any organisms. As concerned the genotypic characterization, which we called as the nucleic acid probes, so first step is you to locate the unique sequence in which the single strand DNA or RNA we use, and then is the generally labeled radioactive or a fluorescent in which the complementary to the sequence of interest, and observe and identify the intact organisms. As concerned the third one, the fluorescence in situ hybridization, which we called as FISH technique, observe and identify the samples, and the last, DNA amplification by PCR. So specific primers are used for this process just to amplify a desired part of the DNA. So as concerned the sequencing of ribosomal RNA gene, which we called as 16S ribosomal RNA for bacteria and 18SR RNA for eukaryotic cells, then evolutionary highly conserved gene variable regions are used to identify an organism, particularly useful in identify the microbes that are difficult to culture. So there are so many other techniques also used, but mostly nowadays the reliable technique is a 16SR RNA technique. So by the combination of the morphological techniques as well as the genetically techniques, we can identify any isolated organisms which we have to use for fermentation process.